Hi all, today I am going to discuss the topic genome organization. This topic will be covered in two parts and today I will be taking the first part. The learning objectives of today's class are understanding the basic concept of genome. There I will be introducing the genome, the genetic material of different organisms, and we compare the genome sizes among organisms. Second, understanding the level of genome organization. Today, I will go over the prokaryotic genome organization, or we'll study bacteria as a model system and we'll learn how the genome is organized in a bacterial cell. So, first I will start with a question. What is genome? Genome is the fancy word for all our genetic content, or genome is the complete set of genetic information in an organism. We can consider genome as an instruction manual which provides instruction for the functioning and maintenance of an organism. The study of genome is called genomics. In almost all organisms, for example, prokaryotes and eukaryotes, DNA is the genetic material. But there are exceptions. We know in viruses, for example, plant viruses like tobacco mosaic virus, or you consider the animal viruses like retroviruses, they have single-stranded RNA as the genetic material. Or you consider the virus which we talk about nowadays, coronavirus, they have a double-stranded RNA as their genetic material. So, viruses may have RNA or DNA as their genetic material, but from prokaryotes towards the higher end, the multicellular eukaryotes all have double-stranded DNA as their genetic material. So, we'll compare the genetic material among these three domains: viruses, prokaryotes and eukaryotes. Viruses may have DNA or RNA as their genetic material that may be double-stranded or single-stranded, and that may occur as circular or linear. In case of prokaryotes, it is always DNA that is double-stranded and circular. Consider the case of eukaryotes. They have DNA as their genetic material, which is double-stranded, but they are linearly arranged over multiple chromosomes. Apart from this, eukaryotes have DNA in their cellular organelles like mitochondria and chloroplast, and they are called mitochondrial DNA and chloroplast DNA. So, now we have an understanding over the genetic material of these three domains: viruses, prokaryotes and eukaryotes. Now we talk more about DNA because it is the stable genetic material across the taxa. DNA is a large molecule comprising two polynucleotide chains. These polynucleotide chains are running antiparallelly. And DNA has a double helical structure in which the two strands wind together to form a ladder-like appearance. The DNA consists of four nitrogenous bases A, T, G and C: adenosine, thymine, guanosine and cytosine. They make up the nitrogenous bases inside a DNA. These nitrogenous bases read DNA like a code which design the function of a DNA. In higher organisms, this DNA is seen localized in chromosomes which are found in the nucleus. For organisms, there are different number of chromosomes. That means different organisms have different number of chromosomes. For example, Humans have 46 chromosomes. If we consider mouse, it has 40 chromosomes, and if we consider Drosophila, it has got 8 chromosomes. So chromosomes differ, and so the genetic content or the DNA also differ among organisms. So far, many genomes have been sequenced. So we have vast repositories of genetic information available in the bioinformatic repositories. So if we explore the genetic content of different organisms, we will be able to compare the genomes of these different organisms. Now let us compare the genome sizes of some of the organisms, some of the model organisms. Organism, chromosome number, genome size, and number of genes. So we'll compare some of the model organisms and their genome size. First, humans. Humans have 46 chromosomes and we have a genome size of about 3.2 billion base pairs. And it contains approximately 30,000 genes. Consider mouse. It has 40 chromosomes. The genome size is 2.6 billion base pairs and approximately 22,000 genes. Consider another model organism, that is Drosophila, which has 8 chromosomes and the genome size is 165 million base pairs and approximately 13,700 genes. Consider unicellular eukaryote yeast genome. The number of chromosomes is 16 and their genome size is 12 million base pair and they possess approximately 6,700 genes. And the prokaryotic cell, for example E. coli, one chromosome, we consider it as single chromosome and the genome size is 4.6 million base pairs and approximately it has 4,600 genes in it. So, we will understand that the genome size differ among organisms and so the genes also, the number of genes also differ among the different organisms. And see, the genome size among all these taxa are very high, very large in size. Now we need to understand how this much big genome is getting packed into simple microscopic cells. Consider the example of humans. In a human cell, we have like 3.2 billion base pair of DNA. And consider if we are stretching out the DNA of a human cell. It says that if we stretch out the whole DNA in a cell, it will come out to 8 foot or 2 meter. That means it is equivalent to a length which is the distance from sun to earth 600 times forth and back. So such a huge DNA is kept inside or packaged inside our microscopic cells. So now we need to understand how the genome is packed inside a cell to fit properly. So we'll look at the genome organization of both prokaryotes and eukaryotes, and at first we'll consider the genome organization in prokaryotes. Prokaryotic genome organization. So we will consider the example of bacteria. Prokaryotic cells are cells which lack a nucleus. So the cells lack a nucleus and they are often very microscopic cells and their cell size ranges from 0.2 micrometer to 10 micrometer in diameter, and their genome size often ranges between 0.6 million base pair to 13 million base pairs. In bacterial cell, the DNA is found dispersed in the cytoplasm in a compacted form, and we call this as nucleoid. So, the genome of bacterial cell we call nucleoid, and that is the DNA seen in the cytoplasm. Consider the example of E. coli cell, which has a genome size of about 4.6 million base pair, and the bacterial cell is around 0.25 micrometer in diameter. Now we have to think this much length DNA, how it is fitting inside a microscopic cell like 0.25 micrometer. So we'll look into the packing of bacterial DNA inside the cell. This could be explained by the model, Rosette model. Rosette model explains the packaging of bacterial DNA inside the cell. So, this packaging or the nucleoid condensation is achieved by forming loop domains and supercoiling. So, the bacterial DNA is highly compacted by these two features by the formation of loop domains and supercoiling. Let us consider the circular double-stranded DNA of a prokaryote. This will be folded, or this will form loops with the help of structural nucleoproteins within the cells. So, they are in a stabilized loop domain formation inside the bacterial cells, and for example like E. coli, there will be 50 to 100 loops, and each loop accommodate about 40,000 base pairs of DNA. So, this will be evident from electron microscopic studies that bacterial DNA is highly folded into a compact structure called loop domains. Now, see, each loop domain are further supercoiled into a much compact or 1,000-fold compact structures. This is called a supercoiling. Each loop get further coiled to form a much compact structure so that the whole DNA will be compacted to 1,000 times. The supercoiling is achieved with the help of an enzyme called topoisomerases. There are two classes of topoisomerase, 1 and 2, of which topoisomerase 2 is responsible for this negative supercoiling seen in bacterial DNA. Now, what is negative supercoiling? Negative supercoiling means the DNA is underwound. That means both the strands can easily be separated out and they will be available for other functions of the cells like replication or transcription. So, in a bacterial cell, the DNA is maintained as a highly folded supercoiled structure which exists in this negative supercoiled coiled nature. And this is achieved with the help of topoisomerase and one more topoisomerase, that is class 1, which help in the relaxation of this walled compact structure. So, together they maintain the tightly packed nature of a bacterial DNA in order to fit inside that cell. So, the prokaryotic genome organization is that the much large DNA is compacted into 1,000 times with the help of this loop domain formation as well as the supercoiling. Hope you all understood the packaging of DNA. Now some facts about the prokaryotic genome. Prokaryotic genome contains DNA, and most of this DNA codes for a protein. So there is very less nonsense DNA in a prokaryotic genome, or most of the DNA of prokaryotic gene codes for protein. Then another fact is that the similar genes or the genes performing similar functions are seen clustered together, or they are regulated by a single regulatory element and are often called as operons. So bacterial genes are often found as operons, and there is much less of non-coding DNA or no nonsense DNA, and prokaryotic gene often lacks introns. We say introns, the intergenic elements are very less in the prokaryotic genome, and the number of repetitive sequences also very less. Repetitive sequence, which is a feature of eukaryotic genomes, which is far less in the case of prokaryotic genome. So that is all about the prokaryotic genome organization, and hope all of you understood today's class. On the next session, we will be taking the eukaryotic genome organization, which is a more complex, compact arrangement, but it is more fascinating. Thank you.